at the National Institutes of Health. In 1993, she joined the faculty at Iowa, where she has been an active clinician, educator, and researcher. She is a nationally known investigator in the molecular mechanisms of antibiotic resistance, and she also runs an active clinical research program focused on infectious diseases. In addition to her role as executive dean, she also currently serves as senior associate dean for the clinical and translational research, as well as professor of internal medicine. So with that, I will hand it over to Dr. Winokur.
Hi, everybody. Thank you for joining. It's 6 o'clock at night, and I appreciate those of you that have taken a little bit of extra time out of the day. It's a hard time to pay attention, and so please stop me at any time if there are questions. I want to make this interactive. They asked me to do about a 30-minute just kind of discussion. And no great surprise, I picked COVID vaccines because it's kind of the topic of the day, the hour, and it's changing minute by minute. So I thought it might be fun for you guys to hear some of this. I'm sure your families are talking about the vaccines, and now you'll have some of the up-to-date information that you can share with them and be kind of superstars when you're at home talking to your family. So I'm going to share my screen. Hopefully you'll let me do that. And now, are you seeing it as one big slide, or are you seeing it as two?
We see one big slide.
Okay, good. Sometimes it likes to divert to the other side of my screens. So I'm just going to tell you a little bit of kind of the background of what's been going on with the COVID vaccine. And we are running a number of the clinical trials here at the University of Iowa. It's been really fun to participate in these trials. You can imagine that people are quite excited to be part of the solution, and it's a pretty interesting story. So let's start with going over just a little bit of the background. And so when you're doing clinical trials, one of the things I like to point out is there are very regimented steps from understanding what's happening in the test tube to actually distributing a vaccine out into the public. And we start with what we call that preclinical phase. And this is the phase where we're starting to understand the drug again in the test tube, but also we're starting to look at animal studies. And animal studies are very important for clinical trials because it's where we first kind of test to see whether it looks like they're working. And also it's our first test of safety. Once we've gotten into animal studies and we're pretty convinced that we're working with a drug or a vaccine that's looking promising and safe, we go into a small number of humans, and that's in a phase 1 trial. And it's usually somewhere around 20 to 80 people that are participating. And we follow everything. We follow EKGs, we follow blood studies, we follow their physical exams and talk to them every day, just to make sure that everything is going correctly. Once we have started to understand that the drug is safe, we're really focused then in the phase 2 of really trying to hone in on the exact dose that we want to pursue. And so that's the phase 2 study, and usually that's somewhere between 100 and 300 people. Again, very focused on safety. We follow things very closely. Once we've understood in the phase 2 what the best dose is, and we're pretty convinced that it's safe in that smaller number of people, we go into a phase 3. And this is usually a big study. Usually 3,000 people are participating in a trial. In that study, we're really starting to hone in on whether the drug does what we think it's supposed to. For a vaccine, that'll certainly be a combination of looking at blood studies to see whether you're making antibodies or some kind of immune response. And also then the key, really what you'd want to know, is it really preventing you from getting an infection? if it's a vaccine. Once we have completed the phase 3, we compile all that data together and we send it to the FDA for them to review and decide if they think that the benefits of this drug outweigh the risks and that this drug is really a good drug for releasing into general use. In phase 4, which is after FDA has approved it, now we're actually out delivering that drug in regular people around the country, and we're often going back and just continuing to follow for safety to make sure that those people are doing what we thought would happen, because now we're starting to get large numbers of people that have gotten this drug and you might see some rare side effects. So that's kind of the standard way that we pursue clinical trials. So now look at COVID. And remember, I think this Operation Warp Speed term was kind of fun in the beginning. You know, I think it got everybody jazzed about, oh, you know, we're really going to pursue this quickly and we're going to get a solution into people and really truncate the COVID pandemic. But I think it came with a price, and that price was as people really started to worry about whether we really had created a safe drug. And so a lot of what we'll talk about tonight is, if your mom and dad ask you, do you think it's safe? You'll be able to give them some of the information of what you think goes into that decision. So under normal circumstances, when we're creating a vaccine, and now we'll kind of focus on vaccines, we find a disease, and I think a really nice example to think about here is Ebola, which is this bottom line here. Ebola was identified in the 1970s, and we spent about 25 years playing with Ebola in the lab, in the test tube, and looking at animal studies. Part of that's because we didn't have a ton of funding. Part of it was, it kind of came and went. It wasn't a disease that looked super dangerous for spreading into a pandemic. After that 25 years of playing with in the lab, we started to create vaccines. Those vaccines were tested in animals and finally then in humans. And after another 10 or 15 years, last year we had the first Ebola vaccines approved. So this is the typical way that we have really pursued vaccines. A lot of it's been, we haven't had a ton of interest from the federal government. There hasn't been a ton of money. Sometimes the disease sputters and it doesn't become a huge national crisis. Now look up at the top here, and what you can see are 3 viruses that we're going to focus on today. SARS was a coronavirus that was identified in the early 2000s. And this was a virus that we started to get really worried about. It spread to about 4 or 5 countries. It was rapidly spread from person to person. People got extremely sick. And we thought that this potentially was going to be the next pandemic. But fortunately, through all sorts of public health measures, they were able to squelch that infection. It didn't spread around the world. We were doing a lot of work on SARS in the test tube around that time. In around 2010, 2012, another coronavirus was identified. This was the MERS virus, Middle Eastern Respiratory Syndrome. Also a virus that caused a pretty rapid pneumonia and people got really sick, and it was spread from person to person. And we started thinking, uh-oh, this one might be the next pandemic. This one didn't prove to be quite as contagious as the first SARS virus was. It sputtered and it's continued to surface on and off in the Middle East. But again, because of that severe illness and spreading from person to person, we started to watch it pretty carefully. And there was a lot of work done in the laboratory between SARS and MERS, trying to understand what parts of the protein, parts of the virus were important for stimulating the immune system, really trying to understand the biology of those viruses. So now we have COVID-19, which is caused by the SARS coronavirus 2. This started right around a year ago, a year plus a couple months. And unfortunately, we didn't luck out the way we did with the first SARS. And this has become a global pandemic. It spread extremely quickly, and people have been extremely ill with this disease. But one of the things that has really helped us is because this is a coronavirus that has a lot of similarities to SARS and MERS, we had a lot of science that had been evaluated in the test tube and in the animals, starting to develop vaccines even to those early coronaviruses. And one of the things that we found when the COVID-19 pandemic started was that COVID-19 shares a lot of similarities to SARS and MERS. And what we know is these coronaviruses all have a very important protein on the outer surface, which is the spike protein. That's the protein that binds to the cell receptors in the human cells and allows the virus to get in. And that is the protein that almost all of us develop really important antibodies to that help prevent the spread of SARS or MERS or SARS coronavirus 2 from cell to cell. Now, these previous coronaviruses, SARS and MERS, we had done a lot of work trying to understand this spike protein. And the luxury of having done all that work for some of those people that really were toiling away on a pretty boring pathogen, you know, when you think about it, NIH was not flooding the coronavirus world with money back in 2000 or 2012. They were toiling away, creating science in their own labs that ended up being so important for what we know today. They even found that these coronavirus spike proteins, that if you mutated these two little prolines right here, that you could change the conformation of the spike protein and make it really more immunogenic. So really able to stimulate the immune system in animals even better. And the good news is when COVID-19 came around, when we sequenced the virus and saw the spike proteins for SARS-CoV-2, it was so similar to those previous coronaviruses. And the scientists immediately went in and made those two mutations in this new coronavirus spike protein. And those proved to be very important as we started testing vaccines. The other thing that you would have seen going on in the past 15 years is not only were we learning about coronaviruses that kind of weren't super interesting for a lot of the years, there were a lot of scientists that were creating new vaccine platforms. And those platforms were designed to be able to create vaccines more quickly. Now, a lot of the work was done because we're starting to learn that cancers can be controlled with vaccines. And so there was a lot of work being done on these vaccine platforms, thinking that we're going to be able to create almost individualized vaccines for individuals that have different cancers. And we'll be able to create all sorts of different ones very quickly. And they were looking at molecular vaccine platforms that were either based on DNA or messenger RNA. Most of the time, we've ended up deciding the DNA vaccines are not terribly great because, first of all, if you remember your cell, you have the cell membrane, the outer membrane, You have the cytoplasm, and then you have the nucleus. The nucleus has the DNA in it. Well, if you're trying to create something out of DNA, you have to get from the cell membrane into the cytoplasm to the nucleus and into the nucleus. So that's a lot of steps for a drug or a vaccine to get into. And it also is the possibility, if you give someone DNA, that it could insert into your own DNA. So most people have kind of dropped the DNA vaccines, but the messenger RNA vaccines have become very interesting. There's been a lot of work done over the past 15 years perfecting the platforms of these messenger RNA vaccines. At the same time, other people were looking at ways to use viruses where you could take out bits and pieces of the original virus and substitute other proteins, sequences, maybe for a spike protein, into that virus. And these are sometimes, adenovirus is the one I'm going to talk about, but there have been a number of other viruses that have been used. And these have started to look very attractive as well, because viruses are really good at getting into cells and making more of themselves and making proteins. So why not harness that? So these are the two vaccine platforms that have really allowed us to make such rapid progress. They're very molecularly based. So all that work from the past 50 years, looking at cloning and PCR and all those things that you've learned about in school, they're now being used to make these vaccines more quickly. The messenger RNA platforms, originally when we were starting to look at those, I was a little bit worried because we had no licensed messenger RNA vaccines. And I thought, FDA is pretty conservative. They're going to want to really see a lot of safety, other things before they'll approve it. But they're very fast to create. Also remember, if you remember from your biology, messenger RNA is very unstable. It breaks down immediately on getting into a warm temperature like a cell. And so they ended up figuring out that they could actually encapsulate that messenger RNA with a little fat bubble. And that fat bubble protected the messenger RNA and allowed the messenger RNA to get into the cells without being broken down. Now, these vaccines have a very big disadvantage, which is they have to be stored at very cold temperatures. Otherwise, over time, they will break down. The viral vectors, the good news about them is we do have a licensed adenovirus-based vaccine, and that was designed for Ebola, which we had approved in 2019. So that was good because FDA likes to use precedent. And you have a precedent now that people have studied these adenoviruses and found them to be safe. They're also fast to create and produce. They're more stable, and they're more stable usually at more typical freezer temperatures, not the ultra cold freezer temperatures. The one thing that can be a problem is adenoviruses are common. And all of us have seen different adenoviruses throughout our lives, and we develop antibodies to those adenoviruses. So you have to be very careful picking the actual type, the subtype of adenovirus that you're using. Otherwise, all those antibodies that you've made over the course of your life might actually reduce the efficacy of the vaccine. So if you look at the messenger RNA vaccines, which are the Pfizer and Moderna vaccines, ones that have been in the news a lot, you can see this RNA is encapsulated in this fat bubble. It's a lipid nanoparticle. That lipid actually helps it fuse with the cell membrane, and it deposits then the RNA into the cell. The great news is RNA is picked up by your ribosomes and translated into protein. And because it's made just the way it would be by a natural infection, it makes a really perfect spike protein. And it exposes that on the surface, and your immune system then comes and recognizes those spikes that are kind of displayed on the outer surface of your muscle cell and creates antibodies. The adenovirus vectors are kind of similar in a way, except for instead of using that lipid bubble, what you're doing is you're putting your piece of RNA or DNA for an adenovirus inside the adenovirus capsid. So adenovirus makes a big shell, and inside it holds all of its genes. And so what you're doing is you're using the adenovirus shell, which is very good at binding to cells and getting the genes into the cells. It dumps the genes in, they go in, again, in this case, it's DNA that's transcribed into RNA, and then the RNA is picked up by your ribosomes, and then that's displayed on the outer surface, and your immune system sees it. So a lot of the work that went into these viruses and vaccines has been done really over the past 20 years. So is it that fast? I think that's the thing that people are forgetting, is we were advantaged by so much science that was done over the years. And that helped us get to vaccines much more quickly. Now, the other thing that happened is because the SARS and MERS vaccines were tested in animals over the past 15 years, we were able to use some of that data to show to the FDA, to show, look, this spike protein that we're dealing with COVID-19 is so similar to SARS and MERS. And we've already got these vaccines that we've tested in mice and monkeys, and it's safe. We should go into human studies a little bit faster. They allowed them to do a little bit of animal work, not quite as much because of that previous work, and they were able to get into human studies, the phase 1 studies, more quickly than they would have if we hadn't had that previous work. But then they did all the same phase 1 studies that you would normally do. They followed people carefully, watched EKGs, did blood work, did physical exams. It was all exactly the same as what we do for any drug or any vaccine. And then they went into the phase 2s where they really picked the best vaccine candidates out of the ones they tested, all being done exactly the same way that we do phase 2 studies for any vaccine. So there weren't steps skipped there. Once we got the phase 2 data, that is when you go into a phase 3 study. And what FDA told the companies is, we're not going to allow you to just show that someone creates antibodies. You're going to have to show us that that vaccine prevents disease. So they told them that any vaccine that they would consider, they're going to have to study 30,000 people to tell if there's a difference between those who got vaccine and those who didn't. And the way the studies were designed is almost every one of these early studies, it was randomized 1-to-1, whereas half the people got active vaccine and half the people got salt water, placebo. You had to have 30,000 people, and most of the vaccines that are being tested today, and we'll talk about the exception, are 2 doses. And then what FDA told them is, okay, this is the kind of basic study design that we're looking for. You're going to have to show us that the vaccine reduces infection, actual infection, at least 50% of the time. You're going to have to show us that these vaccines are safe. 30,000 people you're going to be following, and you're going to be following for every medical event that they have for the next 2 years. Did they have to go to the emergency room? Did they have changes in their blood work? Did they have anything that's different from what they had before they started in the study? And you're going to have to show us some cases of severe coronavirus, because we want to make sure every once in a while there have been some vaccines that have actually exacerbated disease. And so you're going to have to show us enough cases of severe coronavirus 19 to show us that those are happening in the placebo group, not the vaccine group. And then you're going to have to follow everybody for at least 2 months after the full vaccination, so after the second dose, for safety, before we would even consider what we call an emergency use authorization, which is kind of a emergency use authorization is given when there's a public health emergency, like a pandemic, and we don't have the time to watch people for 6 months or a year because the disease is so significant. We will consider a partial authorization for that vaccine after 2 months, after half the people have gotten 2 months after their second dose. I didn't say that very carefully. So that was the guidance from the FDA. The first 2 vaccines that got to the finish line were the messenger RNA vaccines, Pfizer and Moderna. So let's see what they showed. So this is the Pfizer data. So they started enrolling their 30,000 person trial in July. And they got their data to the FDA in late November. And what they showed was almost 95% efficacy. And what does 95% efficacy mean? So they had 169 cases of coronavirus, COVID-19, in the placebo group. They had 9 cases in those that got vaccine. It's a pretty striking difference. 95% efficacy when you do the math. The other thing that was really good to see with this is when you looked at some of these older folks, the vaccine efficacy still looked really good. So that's really important because you guys know that the people who have been most affected by coronavirus have been the elderly. They looked very carefully at side effects. How well is this vaccine tolerated? What we saw was these are vaccines that cause side effects. Many of you have probably had flu vaccines. You don't get a lot of symptoms with flu vaccines. Sore arm, some people, a small number of people will get a little bit of a achy feeling, feel a little under the weather. These vaccines are more what I call reactogenic. About 15% of people will have some sort of fever. The second dose is worse than the first. So you see the first dose, about 4% had fever, and then it went up to 16%. Fatigue was really common, and that was common with both the first and second dose. But most of the time it was pretty mild. You know, most people didn't disrupt their schoolwork the next day or miss work. They didn't disrupt their daily activities. Headache was also one of the really common ones. But there's a really important thing. You remember I said, we really have to prove safety. And I told you typically phase 3 studies study somewhere between 1,000 and 3,000 people. These vaccines, 15,000 people got active vaccine. So that's 5 times the number of people we normally study. What we know with vaccines is the severe side effects that we sometimes see with vaccines usually occur in the first 6 weeks after a vaccine. So we've now studied 15,000 people looking for those really severe side effects, and we didn't see any. So that was really important. And that was the data that allowed the FDA to approve this. Now, the other thing that they got, this is data that wasn't part of what FDA was looking for what they're going to approve, but it's really important data and it's interesting data. This is data looking at antibody titers. So we don't know how many antibodies you need to have to protect you yet. We'll learn that. We're going to learn that. But what we know is here, this black bar, these are the antibody titers in people that have natural infection. So these are people that recovered from COVID-19. Look at this group, the 18 to 64-year-olds. This is the week after their second dose. What we see is they have about 3 or 4 times the number of antibodies that you see after natural infection. So the vaccine is better at creating antibodies than natural infection. So that's a really promising thing. We're going to learn how many of those antibodies you need to protect. We don't know that yet. We'll learn that. But this is showing you that these vaccines are really potent and really effective. And they prevented disease 95% of the time. That's fantastic. And it was safe. We didn't see any serious safety events. We saw that people were a little under the weather for a day or two after the vaccine, but nothing unsafe. So now, last week or the week before, we started to get some of the data for the first adenovirus vaccine in the United States. This is the Johnson & Johnson vaccine, and it's based on adenovirus 26. Now, the thing that was interesting about Johnson & Johnson is they took a risk, and they decided that they were going to test a single dose. So this is great. If you have efficacy with 1 dose, that would be better than 2. In the United States, what they saw was the vaccine efficacy was 72%. Now that's not quite as good as 95%, right? So they had to really think about this. The one thing that they did see is when they looked at their data is it's 72% effective in the United States for preventing symptomatic coronavirus. But if you look at severe disease, it's 85% effective. So FDA is reviewing this data right now. I told you that FDA had said that they would give emergency use authorization if a vaccine is at least 50% protective, and now we're 72% effective. So that's good. Johnson & Johnson also hedged their bets because they knew when they looked at the antibody titers, they weren't quite as good as the antibody titers that were seen after the Pfizer vaccine or the Moderna vaccine. And so immediately after they saw that, they started a 2-dose vaccine study that's enrolling right now. So they kind of hedged their bets. They took the risk and went with 1 dose for their first study, but now they're doing another 30,000 person study with 2 doses. And so I suspect that their efficacy will go up and be better than 72% when they have that second dose, but we'll see. That's my guesstimate. But people were kind of looking at that. If I were trying to convince My parents, do you want the 95% effective vaccine or do you want the 72% effective vaccine? They probably wouldn't be so happy to get the 72% effective vaccine. But this is a really important graph. One of the things that's really neat about this pandemic is all sorts of scientists have made unbelievable contributions. And one of the contributions is all of these people that can do mathematical modeling and model what they think would happen, with the spread of COVID-19. They're modeling now what's going to happen with the variants and how fast they're going to take over. But they modeled what would happen if you had different percentages of vaccine efficacy. And they looked at models of just preventing symptomatic infections. This one looked at averting the number of deaths. And you can see some interesting things. So the good news is Moderna and Pfizer were about 95% effective. If you have a 95% efficacy, you can start to really get into an ability to avert symptomatic infection if you can get somewhere around 45 or 50% of the population vaccinated. If you have a 70% effective vaccine, you have to get a little bit higher. You have to get around 60% of the population vaccinated. So you need more people vaccinated, but you still have a profound effect on reducing infections. If you look at averting deaths, 95% is much better. You have to get 40% of the people and you're getting 90% of the deaths averted. That's great. 70%, you have to get up 60% of the population vaccinated to start to see quite that profound an impact. But when you think about it, even 70% has an unbelievable effect at reducing symptomatic disease and reducing deaths. They've done the same thing with looking at ICU use, hospitalizations, other things. One of the big things we've been so worried about is really protecting the ability to hospitalize people and take care of them. Having a vaccine that's 70% effective and would reduce 85% of hospitalizations would be unbelievably hopeful. So I think we got spoiled a little bit with the efficacy of the messenger RNA vaccines, but the Johnson & Johnson data is really important. And I think there are opportunities to really use that vaccine perhaps differently. So maybe you do choose to vaccinate your elderly population with the most effective vaccine. But we take college kids who tend not to have severe disease and we give them the 70% effective vaccine. That would be really hopeful in preventing spread of disease. That might be something that we'll see coming down the pike. It depends on production and how rapidly we can get vaccine distributed. Other mathematical modelers have looked at what happens when you try and tier who to get vaccine first. And it's a hard decision. We know the people who have been dying. It's been the elderly. It's been some of our underrepresented populations that are in essential jobs. So who do you decide to vaccinate first? That was a hard decision. And there are whole committees that were kind of convened to try and make those decisions. What they made the first choice to do was to vaccinate healthcare providers. And that was to protect the hospitals so that we could take care of patients. And that was knowing that we weren't going to have many vaccines to deliver early on. That was about 21 million people in the United States. Then it got hard. Who do you vaccinate next? Elderly, essential workers, You know, it's kind of nice to have sanitation workers, you know, it's kind of nice to pick up the garbage. It's kind of nice to have electricians when it's -7 degrees out. It's kind of nice to have heat. Those essential workers are key. It's kind of nice to have food. In Iowa, we have an unbelievable food system that goes from the farms to the processing plants to Hy-Vee. Those are really important for the rest of the country. They ended up making the decision to go with kind of a mix in that next phase, which was the elderly and different tiers of essential workers. What's been the problem is when you add those two groups together, it's over half the US population. And we don't have enough vaccine. And so that's why you're seeing this just very difficult situation, people scrambling to try and get vaccines, other things. It's been really hard. And I think that's been the most disappointing thing to see is, you know, we got this vaccine, we got it in record time in a very safe way. Now we would love to have enough doses that we could really start delivering it faster and faster. Things are changing very fast. There are a lot of vaccines. So this was November 3rd to a couple days ago. You can see we went from 14 vaccines that were in phase 2 to 26, 11 vaccines in phase 3 to 20. We've got 4 vaccines that are approved now, 4 have abandoned that they decided to pull out, that they weren't good enough. So it's changing very quickly. What we have coming down the pike, the Johnson & Johnson is the same company as Janssen. That is being reviewed by the FDA right now. We should probably get the results of that in the next 2 weeks. And I'm betting they're going to approve it. AstraZeneca was approved in the European Union. We did not like the quality of that data. They did some kind of interesting mishmashing, especially the United Kingdom's approval. We are actually redoing our trials in the United States, and so we have another 30,000 person trial in the United States, and we're waiting for that data. It should be coming in the next probably 3-4 weeks, I would say, going to FDA. Novavax is a much more traditional vaccine. It's how we make flu proteins for vaccines today. That trial probably is going to finish enrolling this week. We're doing this one at the University of Iowa. We also did the Pfizer trial at the university. So that's going to be probably going to the FDA in April, mid to late April. Another company's got another vaccine. They're gearing up to start their phase 3 trial. So it's just happening over and over. And we need every single one of those vaccines to get those doses so we can vaccinate people as fast as we can. Merck abandoned ship. They didn't like their early data, and they said, well, we're going to let the other companies go. We're not going to waste our money. We have a lot that we're still learning about coronavirus vaccines. You know, we have the safety data from what went into the the emergency use authorization, and now we've vaccinated, you know, probably 40,000 people in the United States with the Moderna and Pfizer vaccines. We saw a little bit of safety stuff with allergies to the vaccines, and I expect that. Vaccines stimulate allergies in people. It's something that we see. So we've learned that. So far, nothing obvious yet in safety. We're watching very carefully to see. we will be trying to understand how long these vaccines are protecting people. We don't know that yet. Whether you'll need to get a booster dose in a year or something, probably. We're learning a lot about the new variants that are starting to arise around the world. We also need to get data in some of what we call special populations, children, pregnant women, cancer patients, things like that. I don't think there's anything that we know that's unsafe about them, but we just don't know enough to prescribe them yet as comfortably as we will after we do the studies. So that's where I'm going to stop, and we can go to questions. What can I answer for you? What do you want to tell your parents now when they're asking you, should they get this vaccine?
I had a question.
Mm-hmm.
So with the vaccines that we have now, like the Moderna and the Pfizer one, is there like a certain one that's suited towards a certain demographic, or do they like both do the same thing?
They're really similar, and I only showed you the Pfizer data because the Moderna data is very, very similar. And so I consider those pretty equivalent.
Okay. And with like the new companies coming out with their vaccines too, do we think there's like maybe some kind of competition to like cater towards the more high risk people or something like that?
So I think that's going to be that question. So Johnson & Johnson only has a 72% efficacy. I think there's a possibility that they may prioritize the most effective vaccines to people who don't mount immune responses quite as well, which are typically the older folks, or maybe cancer patients, someone whose immune system isn't quite as good. What you may end up seeing is differences in allergy issues between the different companies, and that we learn as we go along, and eventually we'll probably be able to say, you have a history of allergies to other vaccines, you're probably going to do better with maybe Novavax or something. That I think could be one of those things that ends up differentiating. I think the less efficacious vaccines like Johnson & Johnson, there may be some recommendations that those should be used in younger, healthier people.
Very interesting. Thank you so much.
You bet. What else can I answer? Are you guys going to get the vaccine if it's offered to you? Anyone saying no? I see all yeses, and the people who've got their screen, or they're got some thumbs up. Anybody afraid? Did I convince you that we've worked pretty hard to make these safe? We really didn't skip steps.
I have a question on the ethics of like the large 30,000 population studies. So when you're splitting groups into like saltwater and potential life-saving vaccine, like the people that might develop serious disease in like the control group, is that just like something that like a necessary evil that like has to be undertaken in order to get the data? Like how do you weigh that when you're trying to do that?
So it was really easy to weigh with the first trials. So we did the Pfizer trial, which was the first trial, and we didn't have anything. So we didn't know whether the vaccine would work. We had no other options for people. And people signed up knowing that they were really contributing to science. They signed up because of altruism. They wanted to help the country. They hoped that they were going to get a vaccine that worked, but there was no guarantee. So that one was pretty easy. It's gotten much harder. So we're enrolling now, and I think we'll finish this week for Novavax. Now we have 2 vaccines that are FDA emergency use authorized. They're not FDA approved, so they're in that intermediate approval. And we know they're super effective. But the problem is people can't get the vaccine. And originally, when we started enrolling, which was really only a month ago, we started with including those that were 65 and older. Midway between the trial, Iowa opened up enrollment authorization to vaccinate 65 and older. And we had a bunch of people that bailed and said, I want to be unblinded and I'm going to go do everything I can to get one of the authorized vaccines. And we wrote into the protocol that they could be unblinded because of that kind of ethical issue, because how can you withhold something from them if they can get it? Now, the problem is they can't easily get it. But we have had people that actually were able to get appointments all over the states. And we've had people coming from all over the state to participate in this study because they really want a vaccine. And the Novavax vaccine in early studies looks really good. Some of them liked it because it wasn't messenger RNA. They thought, oh, this is a more tried and true way that we give vaccines for flu. I like that. But that was an ethical issue. And now as we're going forward, I think it's going to be harder and harder. And so you can change your study design where you're comparing vaccines against each other. And I'm waiting for the time where FDA, I'm hoping soon, says, you know, we're done with the placebo-controlled trials and it's not ethical anymore to do that. And you need to just compare one active vaccine to another. I'm hoping that's coming soon because I think you're right. There is an ethical issue there.
Thank you.
What else can I answer for you?
Hi, I have a quick question. This one's kind of like about your opinion, but how do you feel about like primarily younger people posting pictures of their vaccination cards on social media?
You know, I want people to get vaccinated. And if they got the vaccine because they're, you know, a healthcare student and they're working with patients, that's protecting our most vulnerable people. If they walked by Hy-Vee and they had doses left over and they got a vaccine because they were going to throw it in the trash, I'm good with that. You know, we need to get as many people vaccinated as quickly as possible. And if they could get a vaccine without kind of doing something a little bit nefarious, I'm all for it. And showing your friends that you got it and encouraging them to do it, that's great. I think people feel more comfortable when they see other people have gotten the vaccines and have done well. I think that helps. So that's why we want people to share that experience.
All right, thank you.
What else? How are classes going? Yay. Oh, you got 2 thumbs up, so you guys are happy. Are you bored being online? No? Good for you. Are you waiting for the day that you don't have to be online though? Yeah, okay, that makes more sense. It's a different experience, so hang in there. It'll get better. We're working hard to make it better, so. Anything else?
Dr. Winokur, I had one quick question just about the trial specifically being done at the University of Iowa. From what you guys have seen, has it been easy or difficult to get people to want to participate maybe for the Novavax one now that you guys are doing?
We've had unbelievable interest in our trials and people are coming out of the woodwork. the 65 and older crowd dropped off, and we stopped really trying to enroll them. Initially, we were trying to get about 25% of our subjects 65 and older. And after Iowa opened that population up to get vaccine, we pretty much said, it's too high risk, they're dropping out. And I'd rather get data that the FDA can use. And so it has not been hard, it's actually been very easy. And people are, I've never seen anybody happier than when they have a sore arm, because they know that probably means they got the real deal. And so, and they're really happy if they have a low-grade temp. It's like, yes. So this is one of those times where feeling badly is like a celebrated event. So it's kind of fun.
Thank you.
Okay, guys, have a great rest of the evening. Thank you for taking the time. I know it's late on a Tuesday and you probably have other things that would be fun to do, but it was fun talking to you. And if there's anything I can do or any questions you have, email me.
Thank you so much for your time and all the information, Dr. Winokur. We really enjoyed having you. And thanks to all our members for coming this week. If there are no other questions, we will go ahead and wrap up this event. Stay tuned for our emails. We will be giving you information about our upcoming events. But thank you and have a good night, everyone.
See you later. Bye-bye.
Bye.